Hello there, biology students, and welcome to the second video lesson here for Unit 4, Structure and Function of Cells. The second video lesson, we're going to examine the cell membrane, probably the most important of cellular features and cellular structures. Okay, we'll get into the importance here in a little bit. And then we're going to analyze some of the organelles, some of the other membrane-bound internal compartments that exist in eukaryotic cells, animal and plant cells. So those are the two major video topics for today. 1, the cell membrane, structure and function. 2, organelles of the eukaryotic cell and their structures and functions. Without further ado, let's jump ourselves right in to the cell membrane. The cell membrane is what we like to refer to as a phospholipid bilayer that exhibits selective permeability. So I used a few terms there that you may just be going, have no idea what they may be, okay? Selectively permeable. Let's start with that. The word selective means you get to choose, all right? You get to choose one or the other. So, believe it or not, a cell membrane actually, by some basic foundational principles of chemistry, gets to choose what enters and exits the cell. Permeability means the actual movement, stuff moving through you. So it's selectively permeable, but can choose what goes in and out. Permeability is a word that you should be familiar with from studying some rocks and minerals. We talk about different layers of the earth being permeable, maybe exhibiting some porosity, things being able to enter and exit or flow through it. So the cell membrane is selectively permeable. Another term that may be used to describe it is a regulator. It regulates what enters and exits, and again, it's a basic foundational chemistry that controls it. It doesn't have a brain of its own. It's just the chemistry behind it. And the chemistry behind it is seen with the structure, which is a phospholipid bilayer. Phospholipid bilayer. Breaking the word phospholipid down, there's a phosphate group and 2 fatty acid tails. That little unit right there, that little phospholipid, makes up each side of the cell membrane. Okay? Bilayer, 2 layers. So you have one layer of phospholipid and then another layer of phospholipid. The nonpolar fatty acid tails attract each other in the center, and the phosphate heads are on the outside and innermost areas of the cell membrane. The phosphate heads are polar, and we should recognize that term from Unit 3. So we've got polar heads. The phosphate heads that exist on the outermost and innermost areas of the cell membrane are polar, water-loving, okay? Water-loving. Hydrophilic is another term we use to describe something that's water-loving. So they're polar. Now there are some other molecules that are polar as well, all right? There are some small sugar molecules, some other ions that may be wanting to latch up or get close to those phosphate heads. But there are also some molecules that are nonpolar. Alrighty, now the polarity of the cell membrane is what does a lot of the thinking for it, a lot of the choosing of what enters and what exits. Because again, the outside and innermost, the phosphate heads, are polar, water-loving, and the inner area, those fatty acid tails, are nonpolar. All right, hydrophobic, water-hating. Alrighty, so the decisions that are made by the cell membrane are made by the basic foundational chemistry principle of polarity that we saw in the last unit when we did our little studies over water. Okay, so we've talked about the structure. We know it's a phospholipid bilayer, and we know that the cell membrane can choose what enters and exits. It's a regulator of the cell. So how does it do some of this, though? Like I said earlier, some of it just happens because of the polarity or non-polarity of a substance. And it's a fluid mosaic model. We like to refer to the cell membrane as a fluid mosaic model where every single one of those little phospholipids is moving. It's fluid, okay? So it's kind of like if you were to take 1,000 marbles and cram them into this room with just enough room that each marble is touching each other but can move around. You could take that one marble all the way from the door, all the way to the back of the room, all the way to the front again. Now imagine 2 layers of those marbles in the same action happening. That's what a cell membrane is like. Okay? Constant movement. And because of that constant movement, there's always just enough space, just enough room for certain substances to enter and exit freely. Okay? Some of those substances being water, oxygen, carbon dioxide, but there are some larger substances where it takes a little bit more for them to get through. Alrighty? So again, what's making a decision? The chemistry behind it, the polarity or non-polarity behind the substance and where it's trying to get in through regarding the cell membrane. But those areas open up because the cell membrane is what we refer to as a fluid mosaic model. It's in constant motion, constant movement. It's not rigorous, all right? It doesn't have some type of rigorous structure, okay? No rigidity there, all right? It's always flowing around. Now, there are a few other items about the cell membrane that we need to know. So we've got these things called membrane proteins. These big old proteins that latch themselves inside of the cell membrane where the outer edges are polar and the inner portion of the protein is nonpolar. That way it's attracted to all layers of that phospholipid bilayer and it stays lodged inside of that cell membrane. Now, they move around too, okay? They'll move around too, just like our little marble analogy. All right, they're not staying in one spot. But some of those membrane proteins can do one of a few things. They themselves can act as ion channels where they can let certain materials enter and exit through the cell membrane. Okay, so kind of like a window almost. All right, certain things come in, certain things go out. You get to choose. Okay. They could also act as markers. They could have different types of carbohydrates attached to them on the outside, and they can almost act as kind of like name tags for the cell and assist in cellular communication. They can also be what we call receptor proteins, to where something may latch onto them, and then all of a sudden a chemical reaction begins. A chemical reaction is triggered on the inside of the cell through an enzyme, and some other type of reaction takes place inside of the cell. So a receptor protein is another potential option for a membrane protein, or another potential job, if you will. Okay? And then finally, we can have just enzymes that are lodged inside of the cell, carrying out cellular reactions that may be assisting in the replenishing of cell membrane material, or again, other types of creating or destroying of matter, reorganizing of matter, I guess. Okay, I'm thinking of creating and destroying of bonds. But the reorganization of matter that may need to take place somewhere inside of the cell. So, a few uses of membrane proteins. Okay, that does it for the cell membrane. We should feel comfortable now about the general structure being a phospholipid bilayer of polar phosphate heads on the outer and innermost areas and nonpolar fatty acid tails making up the central portion of the cell membrane. Should also feel comfortable about its major job being a selectively permeable regulator. Controls what goes in and what comes out of the cell. Alrighty? What goes in, what comes out, and because of its structure, it separates the watery internal environment from the watery external environment that the majority of these eukaryotic and prokaryotic cells exist in. Okay? So a fantastic layer that's constantly at work for us, alright, in a various amount of manners. Getting into organelles, we're talking about membrane-bound internal compartments that exist in eukaryotic cells. Remember, this is the major difference between eukaryotes and prokaryotes. Eukaryotes are cells that possess organelles. Organelles, translated, literally means tiny organs. Okay? So we're looking at these itty-bitty little structures inside of a cell that kind of act like the organs that are inside of us, an actual organism. They each have their own specific structure and each their own specific function to assist in sustaining life at the cellular level for that individual cell. So membrane-bound internal compartments is an item that's surrounded in cell membrane. So eukaryotes actually have little structures inside of them that are also surrounded by cell membrane. The entire cell is surrounded by cell membrane. These little structures also surrounded by cell membrane. First one we're going to analyze is the nucleus. A lot of people like to call it the control center of the cell, mainly because it houses the genetic information, the DNA. Now that DNA can be stored in a substance we call chromatin, alrighty, and it tightly coils and condenses at times into the structures that we call chromosomes. Okay? The human body cell possesses 46 chromosomes or 23 pairs of chromosomes. Your sex cells have 23 individual chromosomes, one from each of the pairs. But inside of the nucleus is the DNA the information that your cell needs to end up carrying out all the processes necessary to sustain life at the cellular level. So we call it the control center. DNA is constantly being duplicated, read, spliced, sent around, attached to ribosomes, passed out other places of the cell, and it's constantly being read and translated and turned into other structures, most notably proteins. So it's got all the information, hence why we call it the control center or the brain of the cell. It's surrounded by cell membrane, and we call it the nuclear envelope. And so that DNA can enter and exit the nucleus freely, we've got other structures known as nuclear pores. Okay, little holes, all right, big holes inside of the nuclear membrane so that DNA can enter and exit. Last but not least, the structure that typically contains the condensed DNA is called the nucleolus. Alrighty, a little bit of a substructure inside the nucleus where that genetic information, if it's not being read and turned into something else for cellular purposes, it's hanging out right there. The endoplasmic reticulum is a structure inside of the eukaryotic cell that has two major parts, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The rough endoplasmic reticulum is all about ribosome generation. This is a organelle that is all about the synthesizing, the making of ribosomes, those little protein structures that are in constant work to make other proteins and do other structural, functional jobs throughout the cell. The endoplasmic reticulum is also at times referred to as a transport system, but again, I like to always say how the microtubules and some of the microfilaments and intermediate fibers are the ones that are all about transportation inside the cell, and we'll get to a couple of other items here in a little bit that assist in transportation, but some people like to say that the rough ER and the smooth ER act as transportation organelles for the cell. The rough ER, though, its major purpose, the synthesizing of ribosomes. Now, the smooth ER does a couple other things for us. It's kind of a detoxifier. It's what kind of cleans up a few items inside of the cell. So the smooth ER is a little bit of a filter in a way, constantly detoxifying the cell and keeping the acidity level healthy and happy for everyone, and also getting rid of some other waste and junk. It does another valuable job, and that is the synthesizing of lipids. So it makes fats. Where do we need fats? The cell membrane. Okay? So really important jobs here. Rough ER, making ribosomes, which are the little units that are constantly going around the cell doing little structural, functional jobs. And the smooth ER, detoxifying the cell and also synthesizing lipids. So creating a stock that we can replenish those fatty acid tails inside the cell membrane with at all times. All right? So again, another membrane-bound internal compartment that does a specific job to sustain cellular life. So every now and then, items need to move around the cell in a systematic approach, and items may need to enter and exit the cell in a systematic approach. So we need something that kind of deals with the packaging and distributing of materials that are in constant movement in and or out of the cell, the eukaryotic cell. The organelle for that job is the Golgi Apparatus, also known as the Golgi body or the Golgi complex. The Golgi, I like to refer to as kind of like the UPS man, all right? It's bringing in items, reading and understanding what those items are, and then repackaging them and distributing them where they need to go. It does this with the actual packaging unit being what we call a vesicle. Vesicles are little just membrane-bound balls, packages, in which a lot of cellular items move around in, both inside of the cell and venturing out of the cell. So a vesicle is just a membrane-bound ball. All right? It's just a sphere of cellular membrane that specific cellular items can be housed in, packaged in, and move to and from cells or to and from different organelles within the cell. So like a finished off synthesized ribosome, one of those special proteins, from the rough ER, pinches off the rough ER in a vesicle and then may travel either out of the cell or some other location inside of the cell. The Golgi apparatus does a lot of, again, the reading, repackaging, and redistributing of these materials. That's what the Golgi does. So it deals a lot with the accepting of vesicles, Those vesicles merge with the Golgi, become one. They're reread, repackaged in a vesicle, and then pinch off in a vesicle and travel where they need to travel to. Lysosomes are another entity inside of a eukaryotic cell that kind of deal with the Golgi apparatus. And they linger around the Golgi because, again, the Golgi is that main UPS center. All right? It's kind of reading what's coming into and out of the cell and then kind of kicking it where it needs to go to. Lysosomes are kind of like recyclers for the cell. They are vesicles that contain digestive enzymes. So if there's some waste that's traveling around in the cell, eventually latches up with a Golgi apparatus, okay? That Golgi will just send that waste to the lysosome. That lysosome then breaks down the waste, then latches up with the cell membrane and excretes it out into the external environment. We're talking waste here that cannot just passively transport through the cell membrane. It's too large or it doesn't fit the bill, chemically speaking, all right, regarding polarity or non-polarity. The mitochondrion, the powerhouse of the cell. We call it the powerhouse of the cell because it's kind of like a little refinery, all right? It is the harvester of cellular fuel. Where does it harvest that cell fuel from? Glucose, all right? That amazing organic stored energy that we get by consuming other organisms or raw plant material. So the mitochondrion has a nice little internal network of membranes. Okay, so again, a membrane-bound internal compartment inside of the cell. Alrighty, but it's got more membranes scrunched inside of it where we can have different types of transfer take place. So we want to establish different types of concentration gradients, material we'll get into in the next lesson. But because of its unique structure, the mitochondrion is a fantastic organelle in harvesting energy from glucose. That cell fuel we talk about is known as ATP. So what the mitochondrion does, it is actually, with its internal network of membranes, takes a glucose molecule, breaks it down over several of those membranes, and gives us a very, very, very large amount of ATP per each glucose molecule. It's a very efficient little organelle. And thankfully for the mitochondrion, or thankfully for us, the mitochondrion is the powerhouse. It's constantly providing us with cell fuel, and it's a wonderful, wonderful little guy. So that does it for the general, most popular, if you will, organelles found in eukaryotic cells. The organelles that we just got done discussing exist in both animal and plant cells. But the last topic we're going to look at here are plant cell specific features. Okay? Two of them being organelles and one of them being just a cellular structure. The three plant specific features that we're going to look at are the cell wall, chloroplast, and the central cell vacuole. The cell wall is a feature that is composed of cellulose. So it's a modified carbohydrate. It is just jam-packed full of glucose molecules. It's a very, 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 very large polysaccharide. And because of its unique structure, it assists in giving the cell of a plant cell rigidity. Okay? The cell wall is a fantastic feature for protection, but also structure. All right? Protection and structure. Plant cells need a rigid, structural, functional boundary, and the cell wall provides that for them. The chloroplast is the little organelle inside of the plant cell that accepts the sunlight, the solar energy, and through the process of photosynthesis gives us glucose, that little, most sought-after, organic, stored energy unit on the face of this planet. Chloroplasts have a few other items inside of them. We're not going to get into them now. We'll get into them later in the unit. But the chloroplast is a very, very special organelle inside of plant cells because it's what carries out photosynthesis and takes inorganic forms of energy and transforms them into organic, consumable forms of energy that then other organisms can harvest cell fuel from. The central vacuole, simply put, is a storage unit. And inside of a plant cell, it's got a very important feature. Because plant cells cannot move around because of the rigidity of the cell wall, the central vacuole does a good job of maintaining balance for the rest of the cell, the rest of the cell's liquidy material. So the central vacuole can gather waste, gather ions, gather water, and also distribute waste, distribute ions, and distribute water. So if it gets really, really watery, not to water down the other areas of a cell, The central vacuole inside of a plant cell will actually absorb that water as to not drown other organelle features. If it starts to get a little dry, that central vacuole has stored excess water and can start releasing it to the inner parts of the cell to keep the other organelles in an optimum level of water concentration. So the central vacuole equals storage unit. But there's some important things that it's storing, and there's some important reasons for the storage process. Okay, that does it for this video. Hopefully by the end of this thing, you've got a little more comfort with the cell membrane, its structure and function. All right? Remember, selectively permeable phospholipid bilayer. And also, hopefully you're pretty comfortable with some of the major organelles found in eukaryotic cells, both animal and plant, and with the 3 plant-specific features that we look at with plant cells. You got any questions? By all means, give me a shout. Twitter, email, classroom, phone, text, whatever. All right? See you guys in class.